Buenas tardes, bienvenidas y bienvenidos a una nueva actividad del Festival Puerto de Ideas, Ciencia de Antofagasta 2022, que es presentado por Escondida de HP. Mi nombre es Carolina Torrealba, soy bióloga y a continuación conversaré con el destacado oncólogo austríaco y además cofundador de la compañía BioNtech, Christoph Huber, que se referirá al largo camino recorrido para dar con la crucial tecnología basada en ARN mensajero, que hoy, al verificar su eficacia en el contexto de la pandemia, da paso a realmente esperanzadoras posibilidades para poder generar en un futuro próximo las primeras vacunas contra diferentes tipos de cáncer. Esperamos que disfruten de esta actividad que está siendo transmitida en puertodeideas.cl y en YouTube. Christoph, please take the floor.
So, Carolina and all of you, I'm delighted to be invited to participate, and mostly because we scientists have really the chance to talk to such an audience and to explain what we do, how the different steps are done in order to strengthen the trust into the reliability of science to solve human problems. What I would like to do during the next 25 or so minutes is going with you through our own experience establishing a biotech hub in Mainz, Germany, in the context and close vicinity of one of the big university hospitals, with the aim to produce better drugs, better immune drugs, to treat cancer, to prevent infectious disease and other maladies. I don't know whether this audience is familiar with the fact where innovative drugs come from. It's usually believed that innovative drugs come from big giants, pharma companies. But this, as you see from a fairly old slide, is not the case. The majority of innovative drugs come from university spin-off small and medium biotech companies. This is in particular so in the United States, where meanwhile, this is a 10-year-old slide, about 80% of innovative drugs come from university spin-off companies and are then later on taken up, licensed or bought by big pharma companies. So this kind of changes the focus where innovation comes from. It's in the universities where there is such a great wealth of wonderful new ideas, but where there is not enough competence, resources, and professional skills to move it into the clinical validation, and certainly not into the markets. Some 30 years ago, we have started to establish an immunotherapy hub at the Mainz University Medical Center. Mainz, as you probably know, is in the middle of Germany, close to Frankfurt Airport, where many, many big pharma companies were in the past. Germany was certainly one of the leading nations in producing innovative drugs and companies. At the root, we had a basic research collaborative center funded by the German Research Council, cutting edge innovative basic research, and an international network, research network, where experts from university, from company research, from regulatory agencies met, exchanged ideas how basic research can be translated. And that was one of the powerhouses, together with the Collaborative Research Center and the first tumor vaccination center, probably worldwide, but certainly in Europe. That was the source of the ideas, using the power of the body's immune defense to fight diseases or to prevent diseases. Specifically, we addressed questions of antigens against which one can produce immune drugs like vaccines, like therapeutic antibodies, or genetically modified immune cells. Ideas how to personalize that were developed, and certainly all the cutting-edge technologies required to do so, including next-generation sequencing, bioinformatics, digitalization, and robotics to speed up and improve manufacturing were included. I was certainly the oldest who started with the academic background. And then after 10 years, I found two wonderful and now the leading scientists and leaders in BioNtech and in other of our organizations. Ugo Shahin, who really is a technology genius and a wonderful leader with passion and vision and extremely good to teach young students and scientists. And Özlem Türeci, his wife and partner since the very beginning of his research, who is so extremely competent to implement these ideas into clinical studies and validation and even approval. So we started together 20 years ago, and they are the operational leaders. And most of the ideas can come from Uber. The components, when that kind of matured over the first 10 years, so between 1990 and 2000, was we understood we cannot cross the gap between university and professional manufacturing and clinical testing without founding companies. This, because there you find perhaps the financial resources, there you find the competence to manufacture drugs, there you find the professional skills to do the regulatory approval, to test and perhaps even approve drugs, among many other things. We had a number of companies, spin-off companies, and you know about BioNtech, which is by far the biggest one, which we have founded in 2008 under Ugo Shahin's lead as CEO. We understood that the gap between university and its culture and professional commercial organizations is a deep one. To cross that, we have funded another organization as a non-profit company, kind of spin-off from university, to improve the professional performance and to speed up and improve the output at Translational Oncology Institute, TRON. There, they developed platform technologies. That they did develop all the services like cutting-edge next generation sequencing to unravel the genome of tumor cells or viruses and whatever. And the third kind of organization we have incorporated and founded into the immunotherapy hub was a cluster funded by the German federal government, in which you kind of learned how to partner with about 100 organizations, both non-profit, university, or companies. And that kind of formed the momentum in which one really could jump high hurdles. Huber's probably most successful project was using messenger RNA, which, as you know, is a highly unstable carrier, a kind of master plan of information to manufacture proteins. And proteins, as you know, is life. This has been done by many, but the problem to make it effective is a threefold thing. First is the information must be delivered to the right cell when it's injected into the patient's body. The right cell to do a vaccine and to take up mRNA is the so-called APC, the antigen-presenting cells. This is the load of the adaptive immune system, which makes antibodies against tumor cells, against viruses. That adaptive immune system makes killer cells tumor-specific, virus-specific, to eradicate, for instance, virus-infected or tumor cells. mRNA, and that has been uncovered by Augustine, is specifically and selectively taken up by antigen-presenting cells. And suddenly the entire adaptive immune system starts to work. The problem is that mRNA is very unstable. It doesn't make sense for a cell to accumulate multiple carriers of information for whatever protein is produced by a cell at a different time point of the cell life and in different functional states. So one must stabilize, and don't read that. It just should give you an idea that all genetic maneuvers have been applied to improve stability. And many have done that, but Huber's constructs work extremely well. And what you see on the right side, the potency to induce and activate immune cells by the Huber modified mRNA became multiplied by something like 5,000 to now about 50,000 times. Suddenly, mRNA became a drug which is capable to induce protein formation in the body when injected. The third is, it has been found out when you encapsulate the mRNA into what has been called nanoparticles, very, very tiny small particles, nanometers in diameter. They protect the mRNA further and stabilize, and they enter the antigen-presenting cells much better. So this was the prerequisite to make a drug. And from that drug, scientists at BioNtech, Huber and Katikarico, who is also in the public media attention, very rightful, have established two different mRNA formats. One is the unmodified uridine mRNA, and this turned out to be extremely good for tumor vaccines. And we'll show you examples of that in the next few slides. This because they induce very strong so-called CD8 killer cells, which kind of kill tumor cells when loaded with tumor antigens. And the other is a nucleoside-modified mRNA, which is much better to induce very strong neutralizing antibodies. And that turned out to be the format of choice of the first COVID-19 vaccine in the past. So the advantages of mRNA vaccines are probably fivefold. It's selectively taken up by the load, which kind of sets the immune system in motion. It does not integrate into DNA. It does not change our genetic information. It does not require further adjuvants to increase the potency of a vaccine, which most other vaccines, conventional types, do need. It's a simple manufacturing process, and so far in most of the well-studied application, it is superior in efficacy to the conventional formats. So we started with cancer vaccines about 20 years ago when Huber started in the collaborative research center context, and then more than 14 years under the roof of BioNtech and under its leadership. We'll show you examples how that worked. Tumor-associated antigens are the only way to design rational and effective tumor vaccines. Let's look back where they have been discovered. Tumor antigens, which can be used for vaccination, have been first discovered by Thierry Brun and molecularly unraveled at the Ludwig Cancer Research Institute in Brussels. And antibody recognized antigens by Hugo in his previous university life. There were two groups of antigens, tumor-associated selectively expressed antigens not expressed on normal cells. One are so-called shared antigens, which are shared between tumor cells and, for instance, embryonal normal cells, and re-expressed when a cell transforms into a malignant cell. And the other are neoantigens coded for mutations. You all know that cancer is caused by mutations. And these mutations are new and are not known to the immune system of the patient. Against these antigens, you can also vaccinate. So we have two types of mRNA-based vaccines moved into clinical testing over the last almost 10 years. One we call FixVac, is the combination of 5 shared antigens, which kind of are specific for every different tumor entity. They come from the shelf. This is a comparatively simple process, and that is very effective in the first clinical testing in advanced melanoma patients who did not respond to any type of medical treatment any longer. You see, almost 1/2 of the patient, and this is the maximum diameter of each patient's tumor subsequent to treatment with such FixVac vaccines. And you see that about 1/2 increase and 1/2, as one would predict in a refractory disease, and one half regress. And this is frequently very stable. So that was beyond expectation. The other is technically and perspective-wise much more interesting. We call that iNest. These are fully individualized mRNA vaccines, where every patient, tumor and normal cell, is sequenced with next generation sequencing. Mutations are identified by comparing normal cell sequences with cancer sequences. And since there are about 100 mutations in each, on average in each tumor, and since these mutations are individual, unique for every single patient, you have to do that kind of assessment as well as the manufacturing of a vaccine in a tailor-made fashion. And that has to be done fast, otherwise the patient is not going to survive. And it has to be done in an affordable, feasible manner, which means it can't be too expensive. This can only be accomplished by applying digitalization and robotic technologies. We have 2 such factories established now, a third one in the process of approval, where we can do that process in a feasible manner within less than 4 weeks. And we have hundreds of patients treated meanwhile. And you see here the results of our first study, which is now something like 7 years old. This are the frequency of relapses of that small group of patients during the last 2 years prior to treatment. And the green dots represent the relapses after treatment. So each patient is her or his own control. And these are the survival rates. And you can extend that line for almost 7 years. We don't see the relapses after something like a year. This is very remarkable. And we do that now on large numbers of patients together with the former giant, BioNtech, Roche. And we'll see whether we can really cure these early patients who otherwise are going to relapse. Another thing which more recently has been established associates with the adaptive transfer of genetically engineered T cells, immune cells of patients. You take out immune cells from the bloodstream, you transfect them with a receptor, a chimeric receptor, which I'm not going to explain what it is, which recognizes tumor antigens on the surface of refractory diseases. The first clinically approved CAR T cell receptor against the CD19 antigen was developed by my friend Carl June at the University of Pennsylvania Medical Center, cured and is now meanwhile approved on large scale about 70% of refractory children and adults with acute lymphocytic leukemia, and a similar proportion of refractory malignant lymphoma patients carrying that specific antigen. And this was the first child which has been rescued by that therapy. We have developed a similar technique in solid cancer using our proprietary Claudin-6 antigen and using fairly small numbers of adaptively transferred genetically modified T cells, which then have been expanded in vivo by the concomitant application of a vaccine directed against the same antigen. And what you see here is that the frequency of these genetically modified cells in patients prior to vaccination, and you see a sharp increase, several orders of magnitude. So they are expanded in vivo. This is cheaper, this is less toxic, and one also sees dramatic responses in about 45% of the phase 1 patients. So you see many cancer applications, infectious disease, and I'm going to go through that very quickly. You're all aware of the COVID-19 vaccine project. The unique thing is the light speed without violating any regulatory rules. It is again a Huber's thing, both in terms of the design of the vaccine and in terms of leading the BioNtech group through the process and to instruct and motivate the big pharma partner Pfizer, who is a 50-50 partner in that development, where the intellectual property is owned and still owned, and the technology also by BioNtech. As you know, it addresses the spike protein. If you successfully raise antibodies or virus-specific killer cells, you certainly prevent or eliminate disease much faster. You know about the speedy process. Within 11 months, one got approval, and that was beyond expectation for everybody, because usually it takes 5 to 10 years to develop with conventional technologies such a procedure. This has been made possible, as we believe, by the mRNA technology itself, which, as you have seen, is a speedy process, comparatively simple in terms of the mRNA component, very sophisticated in terms of the packaging into nanoparticles. It has been caused by finding an ideal big pharma partner who was very competent to do large-scale clinical studies with more than 40,000 individuals to get approval, which we small ones would never have been able to accomplish within such a short period of time. It was fantastic as well. Then we were used to make fast vaccines by the cancer immunotherapy, the individual vaccines. And certainly the regulatory agencies supported that. In a different way, Europe took a little longer, but what are a couple of weeks in such a process, which usually takes forever. So we have to be grateful for the support. And we also have to be grateful for the strategic investors, which kind of enabled BioNtech and agreed to go such a high-risk project, which, if it had failed, would have certainly killed the company. You know, the early effects were fantastic. And you all know, we have learned that the protection doesn't last forever. 3 to 4 months. Revaccination kind of re-establishes protection. And right now it looks like that we can do 4 cycles and continue to protect individuals from serious disease. We do not know how that will go. I will show you a national example from my own home country. This is one of the few institutions guided by Eva Hölzel, one of the big Austrian banks with more than 10,000 employees, where they have a more than 90% vaccination rate. And you see, the longer you vaccinate, the better the difference between the average national, which is very embarrassing in comparison to what you have meanwhile accomplished, and the vaccinated population. And that increases, the difference increases almost every week. Let me very briefly address some of the very prominent questions, but not in a granular fashion. There are Omicron vaccines now in clinical testing, and we and others believe that within the next few months, in agreement with the regulatory agencies on both sides of the Atlantic and multiple nations, and countries will get it to clinical large-scale application. And then we'll certainly learn during the next few weeks and months whether that is better and whether we can improve the first vaccine's efficacy. Obviously, second generation vaccines are developed, not only using S1, but also a number of other viral proteins. And that is going to be seen again in clinical validation studies. Let me say a few words about variant viruses. We're all aware of the mutant viruses, how they emerge, how difficult it is to predict the clinical efficacy of vaccines without having epidemiologic endpoints of frequency of infections and their severity. It cannot be predicted from the sequences, and it certainly cannot be predicted without clinical observation. And that makes it difficult because experts express their opinion, and they are sometimes right and sometimes not right, as long as we don't have facts. But you all should know the facts will be established and rational decisions will be made on that basis, not on the prediction basis. A word to solidarity. I can certainly not speak for all the other companies in the area, but I certainly can say for BioNtech and for our partner Pfizer that solidarity with the poor one certainly is a priority. Right now, much more than 50% of the biotech probably contributes something like close to 2 billion. More than one half is exported. With the approval of the European community, we are still under pandemic rules to poor and medium income countries, and usually sold without any profit. The idea is now to establish, and the first so-called BioNTech are already delivered, manufacturing units, container type, which will be spread all over the globe and will then be accessible to organizations, to countries, to make their own vaccines under support and guidance by the company. This is now the new concept established by Secretary COO of BioNtech. And we also want to incorporate that into a partner-like structure to fight pandemia and to enable regional production of other innovative drugs, like, for instance, genetically modified CAR-T type of cells to treat malignant disease and a number of other things. So, in a nutshell, I'll skip that. We certainly believe into the power of science to solve problems. The mRNA technologies are validated now for the first time in one indication, and that certainly supports and enables to go into multiple other indications in cancer, in autoimmune disease and allergy, and a number of others. For instance, replacing proteins, defective proteins in hereditary disease without doing gene therapy, integrating therapeutic genes into the DNA of patients. And what we also have learned, that synergy, not domination, of such a development process really helps to advance innovation. So we have synergy between public, like universities, or the Tran Institute, which I have mentioned in the beginning, and private company type of organizations. We took advantage, and others too, between in synergy between big and small companies, and also across the Atlantic between different countries, and that is kind of wonderful. And we don't hide; we share data. We work to solve one problem we all suffer from, and that kind of opened a new dimension of translating science into survival. It must be said that this is only possible and certainly carried by people. And we need to find the talents to support them, to protect them, and to help them to translate their visions. And that is kind of another thing which we hope will enter the culture of all our countries. More belief into science to solve all those problems which we have, and we have many in these days. So thank you very much for being present.
Thank you very much, Christoph. It was a fascinating talk, fascinating concepts you mentioned. I need to start just saying thank you for helping developing, not only for your talk, but also for helping to develop this with you and all the collaborators to help develop this technology. That's the basic of a vaccine that a lot of millions of Chileans have received, and has helped us to combat this awful pandemic that has cost a lot of lives and has transformed people's lives. So thank you very much for that. It's really touching to see all the work you have led with, and know with a lot of institutions and more people, but finally has such a concrete, effect on our lives. So thank you for that. That's a pleasure.
Thank you. And you understood it's not me. I'm just one of this. I'm certainly the oldest and the one who didn't do the operational burden, which is so essential, as you know.
You're part of this change that we're going to be talking about. So the first thing I wanted to ask you, it's like a very general question, but I want people to really understand the innovative and very innovative part of what you have just said. So you said 20 years ago you were start working with these cancer vaccines. So I think that's really amazing because 20 years ago, I don't know if people really look at you as if you and your collaborators were crazy, but in the field, you know, it was not something very obvious that you should, you know, work with the immune system to treat cancer. And as we have just learned because of this pandemic, and we learn at school, but also because of this pandemic, we've learned that the immune system actually attacks pathogens, you know, external things that get into our body. And you are telling us in this talk that all the strategy built in the last 20 years have been used, that immune system that we've trained to detect external agents to actually treat cancer, which is an illness that it's part of our body. It uses our own cells, our own proteins. So that was very innovative at that time, and still a very, very new way, you know, to look at cancer, you know, to use our immune system to treat something that it's our own body. I guess people was at the beginning very scared about you maybe triggering some autoimmunity or something like that, but I just want you to comment on that. So how all the things that you're telling us, it's like this innovative strategy is to use our own immune system to actually treat cancer cells, as you explained, because identifying these very different antigens in tumor cells that can be attacked, and how innovative was at that way. I'm really curious about people thinking that you were all crazy, and how has that changed today, that it's actually something that it's going to be. And maybe the next question is how long do you think it takes for this technology to actually reach to patients massively?
Well, thank you, Caroline, for asking that question. And I understand you probably should teach rather than me. You understood very well the concept. So, some 30 years ago, we were very few around the globe to believe into such a concept. And at that time, in the early 90s, the molecular definition of tumor antigens, which can be used to vaccinate against, have been formally proven. And that was kind of the door opener to the further concepts. The word out in the street still is that tumor vaccines probably don't work very well, or probably don't work at all. But every year, we and others accumulate evidence in terms of clinical data, which prove beyond any doubt that one can successfully treat, and perhaps cure patients with that technology. That also implies that we have to improve the technologies further, and that we have to show formal, convincing evidence that the regulatory agencies will allow us to enter the market with the products. We would hope that the first tumor vaccines will be approved in the next years, but I have not the skills to look into the crystal ball. But prediction is, it is likely.
It's not very scientific, probably, to predict.
Well, the burden of proof is to do multiple large studies in different indications. And that is what happens right now. Mm-hmm. During the early COVID pandemic, that work had to be stopped because almost every test center closed down So no new persons could be treated. And within BioNtech, about 1/2 of the employees decided to be moved into the COVID-19 vaccine development in order to speed up the process. Now they returned back and they do the former oncology and other studies. So we do hope that it won't last too long.
We're going to go to the COVID vaccine later, but after turning to that, is this guess something that makes sense? So if we are using our own immune system to actually attack some specific cells, maybe you could guess that the other effects that when people live with cancer have to take these drugs that attack not only cancer cells, but the rest of the cells, having these very hard effects to live with. So would you guess that using this technology, maybe the secondary effects are milder, or have you seen something like that? Does it make sense because of the theory that you're using no external drugs, but your own immune system, that could be much more precise, getting to that cancer target?
Again, it does make sense, and you kind of found the right word. It is more specific than chemotherapy. It does usually not affect normal cells, and therefore it kind of satisfies the patients and the health professionals' hope to find better tolerated treatments. And so far, this is the experience that we and others have. The same seemed to apply for the genetically modified CAR T cells, but there it's a still ongoing phase 1, and the data are not ready for final publication. But right now, side effects are very tolerable with the low cell dose and the vaccination, which then leads to in vivo expansion of these angry killer cells to kill the tumor. So it's a new dimension to treat. It's a new thinking behind. It is very specific. It requires other diagnostic procedures, and it is highly personalized.
So we really need all the people involved that we hope that can be very close to reach to the patients in a more massive way. It sounds something that we hope that really can happen very soon. So I wanted to go to your experience also, because you have been a privileged, actor of seeing all this process going through. So first, I wanted to ask you about your experience. How have you seen through this pandemic that has been also, and with all the respect to the work and the process that we have lived, also kind of an experiment in terms that it has accelerated a lot, or it has united people to work on the same problem, all the world working on the same problem. And that was also kind of a global experiment to really have vaccine, as you mentioned, in 11 months. That was like crazy. It was like no way we could have thought of that, having this sequence and then crystallize the proteins and all that in such a short time, and then you people developing this vaccine, and it has been crazy. And all the world in science also changed the way they moved, changed the way the knowledge moved. It moved faster, it moved like more strategically. All the people worked together, people that didn't talk really started to collaborate. And so how have you lived that? And especially, like, do you think all that you mentioned, the synergy, the data collaboration, all that, do you think that some of that are going to change the way science and drug development normally works? Or if this is going to be like a parenthesis and then we're going to forget about it and we're going to keep working the way we were working before pandemic? So do you think we can really innovate also in the way we produce knowledge, in the way we move together to make big changes, in the way we approach to big challenges, as science have really shown to the world, very different to what has happened in politics and the rest of the world. You know, science has been really something that we have admired, the way it has reacted to this huge problem.
Well, again, thank you for discussing that point. I think that is beyond any doubt that every organization and every single individual involved, whatever the contribution was in that process, has learned how things can be done better. And everybody learned that synergy and collaboration can enable to jump hurdles which before have not been crossed in a short period of time. So it will have consequences. Political leaders think in these terms. Science organization think in these terms. The general public has seen that it is possible and will ask for that when we are confronted with similar problems. So it has already had an impact, and that will be long-lasting. The other thing is the overall recognition of science and innovation to improve our life. If you think back, evolution of mankind, innovations have changed the life and certainly made it better. Fire, water power, electricity, you name it. Vaccination was among the most successful medical interventions in medicine. Every fourth died from infectious diseases. And now, except the pandemic, very few die. It has been scientifically extremely well developed. We have seen heroes like Pasteur, Robert Koch, and the many others to show how science can remove the threat of disease like smallpox, which killed 30% of the population within weeks. So now we have found a new way to revitalize and to further expand the vaccination medicine. We, on the other hand, are faced with the problem that the discussion has changed from a scientific and medicine-driven discussion into a discussion about ethics, which is okay, and fake news and other unexpected developments. So society is challenged with the problem how to educate people. to increase the trust into science and to make them understand how fake can be discriminated from fact-based experiments and knowledge. And that is a long way to go. And that will involve schools, first of all, and all our households. And this is a public discussion going on, which certainly cannot really be led by the scientists and companies producing. This is the challenge of the society, and this will determine how we handle innovation and whether these positive experiences we have made over the last 2 years can be at a large scale translated into the solution of other problems. And we do have so many. Think about these apocalyptic horse riders we have beyond pandemic, what we have in climate change, that calls for science solutions. Think about cybercrime. This calls for science solutions and ethical legal. Think about migration. On a science-based concept, one should be able to accommodate the poor ones and those who are driven out of their countries, to feed them and make them happy to stay.
Yeah, so
sorry for preaching, but it's
it's super
very
interesting.
close to
yes,
my heart.
it is super interesting. One of the things that I kept thinking, like since the year that the pandemic started in 2020, was actually that, like, we went through this social crisis in 2019, and then pandemic started, and even in that social environment that was super difficult to trust, even in that context, all the field of science, and I actually had the opportunity to lead a project where all the people, really lots of universities and lots of researchers work together in order to give solutions to our country. So I kept thinking what you say. So how can we kind of replicate this experiment for other, challenges that we have, like climate change, as you said, in our country. Our country is actually going to be one of the countries that had more effects of the climate crisis. So we have a lot of things to solve on that point. And taking that, because as you said, they're going to be this experience is going to last. It's going to have, long-lasting effects in the way that organizations cooperate, etc. So can you talk a little bit more on that? So how do you think really are going, or how do you see they are going to change? Especially, I want to ask you this question because you, Christoph, you're a very rare specimen in the world.
Because of my age.
No, that's not strange. But you, as you were telling me before we started, you're an MD, you treat patients, you have treated patients, you have been a university professor, scientist, a very well-known researcher, but also you have participated in biotechs, also you participate in big pharma. And you've been an actor of all the stages, maybe, of the creation of knowledge to reaching to the patients, and with this problem that we have in the society, that scientists tend to be very well recognized, and people trust in scientists, but they do not trust equally in big pharma. People don't see this as a chain. So can you tell us, from your perspective, that you've been an actor of this, of all the stages of this change, how this chain works, and why it's important for countries like our country to really promote not only one piece of the change, but maybe the whole system, or it's not that way? What do you think about that? How can we, and in the context of what we have experienced in the pandemic, so how are these steps of the route are going to be changing, how it works, and what can we do to really promote a very virtuous ecosystem that can help us to attack and address big challenges?
Very complex question.
It's the last one.
Very simple answer. Put the patient in the center. This is it. Be aware that is a chain involving universities, small, medium-sized enterprises, big pharma, regulatory agencies. If you see that as one chain with individual contributions to advance to the next step, and if always the patient's interest is put in the focus, then this will work. If you put ego, monetarian interests, or whatever besides in the focus, it will work less. So it involves a clear definition of priorities, a clear definition of the places and contributions. And the society should become aware of that multiple steps and multiple institutions are required. It's not big pharma alone. Never ever. It's not the scientist alone. And it goes step by step. If they interact properly, wonderful. There's another thing which you certainly are aware of, and that is who pays for the development of innovative drugs, at which stage. We usually need 10 to 15 years to approve a biologically innovative drug. We usually need 1 to 3 billion range US dollars, something like that. An unbelievable high price. Who is going to pay for that prior to marketing? And being aware that so many fail prior to really get revenues by approval on the market. And the answer is every component of the chain. Universities on the early spin-offs, financed usually by venture capital, the intermediate, and then big pharma usually, or very successful small, medium-sized enterprises who kind of grow into a pharma giant are the ones in the more final steps of approval. The most vulnerable step is the venture capital in terms of the finances. The venture capital financed small, medium-sized enterprises, because there is a different acceptance of high risk in different cultures. We know that in the United States, venture capital per capita is about 7 to 8 times more than in Europe. 7 to 8 times more. Can you compensate that by smartness, dedication? Sometimes, but it's very, very hard. Can you improve and educate to become less risk aware? Probably. But it's again a question of education. And again, a question of understanding who pays for what at which stage. Can governments do something? Sure. They can, for their highly relevant venture capital investments, highly relevant for society, certainly apply tax reduction. That certainly would motivate private investments into it. But one must be aware that the intermediate step of development of innovative drugs is mostly financed by private venture capital, willing to accept the risk of failure. And if one can reduce the hurdle and increase the willingness to accept, then a society will benefit. And it has nothing to do with greed. This is what happens in real life. And this has more to do with motivation and not so much with earning money. But that is not understood. And that is again something we need to educate. And the politicians will then be willing to kind of favor those things which are relevant for society. So I do see that this kind of lesson has also partly been learned during the process of pandemia, but it's by far not complete. And one needs to work on that.
Thank you, Christoph, really very much for this talk, this conversation. And I really hope you can come some next time. It would be really nice to have you here and extend the conversation. But now we are going to finish this conversation. So thank you very much. And for your time, for sharing your experience with us and for inspiring us with all your knowledge and ideas.
Well, thank you very much, Caroline. It has been very enjoyable and profitable talking to you, and I certainly would hope that we can meet in a face-to-face. And thank you, Sergio and Jose, for making this possible. I have no idea whether that kind of will be useful for the Science Festival's mission, but I really hope that at least it can contribute to convey what you plan. Thank you very much.
Muchas gracias por acompañarnos a todas las personas que han estado en esta charla. Les invitamos, por supuesto, a seguir disfrutando de las actividades del Festival de Ciencia Puerto de Ideas Antofagasta 2022, que se va a desarrollar hasta el día 24 de abril, y pueden seguir revisando, por supuesto, la programación del festival y todo el contenido de Puerto de Ideas en la página web puertoideas.cl. Chao.